Unit 2 Lipids and Proteins MFN-002 Nutritional Biochemistry First Year Master of Science, Food Nutrition, MSCDFSM, Revised Master's Degree Programs, School of Continuing Education, SOCE, IGNOU audiobooks #IGNOU audiobooks Indira Gandhi National Open University 2.1 Introduction. In the last unit you were provided with a broad introduction to metabolic and nutritional aspects of biochemistry. The chemistry of carbohydrates, one of the physiologically important molecule, was focused next. In this unit, we continue with our study of important molecules by focusing on lipids and proteins. Major topics include structure, function and metabolism of amino acids and lipids. We would like to remind you once again that this unit is the basis for understanding the concepts related to lipids and amino acids, proteins as discussed in the Advanced Nutrition and Food Science courses. Hence, it would be a good idea to go through the units on lipids and proteins in these courses together, as the information in each course would supplement each other. Objectives: after studying this unit, you will be able to describe the structure of lipids and classify them, explain the chemical properties of fatty acids, neutral fats, phospholipids, steroids and eicosanoids, illustrate the structure of amino acids, peptides, proteins and nucleic acids, classify amino acids, peptides, proteins and nucleic acids, and describe the chemistry of proteins and nucleic acids viz. Amino acids, peptides, proteins, nucleotides and nucleic acids. 2.2 Chemistry of lipids. Introduction. Lipids are another important group of nutrients that should be looked into with interest by a learner of dietetics very seriously. This subsection intends to focus on certain important aspects of lipid chemistry. These are as follows: chemical nature of lipids, major classes of lipids, important lipids of each class, properties of fatty acids and neutral fats. At the outset, let us get introduced to lipids. Lipids are a heterogeneous group of compounds occurring in both plants and animals. These are insoluble in water, but soluble in other solvents such as ether, chloroform and benzene. Because of heterogeneous nature, it is difficult to define these compounds in certain terms. However, neutral fats and oils derived from animal and plant sources are called neutral lipids, which on hydrolysis yield glycerol and fatty acid. We shall learn about the classification of lipids next. 2.3 Lipids. Structure and classification. Lipids are classified on the basis of their chemical structure as presented in Figure 2.1. Look at the screen. Figure 2.1. Classification of lipids. Simple lipids are esters of fatty acids with various alcohols. They include neutral fats, which you learnt earlier, are esters of fatty acid with glycerol, and waxes, which are fatty acid with alcohol other than glycerol. Compound lipids are esters of fatty acids containing groups in addition to an alcohol and a fatty acid. Examples include phospholipids, containing in addition to fatty acids and an alcohol, a phosphoric acid residue, glycolipids, compounds of fatty acids with carbohydrate, containing nitrogen but no phosphoric acid, etc. Derived lipids are substances derived from above groups by hydrolysis. This group includes fatty acids, glycerol, steroids, alcohols, sterols, fatty aldehydes and ketone bodies, vitamin A, D, A and K, etc. In the following section, we shall get to know a bit more about these lipids. A detailed discussion on all lipids is not included herewith, however, some important lipids are focused. We start our discussion with the chemistry of fatty acids. 2.3.1 Fatty acids, saturated and unsaturated. We saw earlier that fatty acids are obtained by the hydrolysis of neutral fats. Many different fatty acids occur naturally in foods. In their free form, the fatty acids have the configuration shown herewith. Figure 2.2. Fatty acids where R is a hydrocarbon chain, CH3, CH2, CH2. Fatty acids are grouped into two classes, saturated and unsaturated fatty acids. The terms saturated and unsaturated fatty acid may not be new to you. Every other day there is one report or another, highlighting the useful and or harmful effects of these fatty acids on health. What are these fatty acids? When a fatty acid contains one or more double bonds, C=C, it is said to be unsaturated, otherwise it is a saturated fatty acid, as highlighted in Figure 2.3. Look at the screen. Figure 2.3. Saturated and unsaturated fatty acid. Presence of double bond in a fatty acid makes it relatively unstable to air and suitable reagents. Physical properties of the fat are also affected due to the presence of double bonds in the fatty acids. Fats rich in unsaturated fatty acids usually exist as liquid, i.e. oil at room temperature, while fats rich in saturated fatty acids exist in solid form. All food fats contain both saturated and unsaturated fatty acids in varying proportions. A detailed discussion on saturated and unsaturated fatty acids follows. Saturated fatty acids. General formula of saturated fatty acids is CH3, CH2, NCOOH. It has a polar end with a free COOH group and a non-polar end with a hydrocarbon chain, as can be seen in the structure of fatty acid illustrated in figure 2.2. Fatty acids with less carbon atoms are readily miscible with water and the solubility decreases with an increase in carbon atoms. Saturated fatty acids with less than 10 carbon atoms are liquid at room temperature and those having more are solids. Melting point of these fatty acids increases with increasing number of carbon atoms. Table 2.1 lists the common saturated fatty acids of food fats. Table 2.1 Naturally occurring common saturated fatty acids. Common name, molecular formula, occurrence: butyric acid C3H7, Cu-butter caproic acid C5H11, Cu-butter caprylic acid C7H15, Cu-fats of plant origin capric acid C9H19, Cu-fats of plant origin lauric acid C11H23, Cu-palm kernel, cinnamon, coconut oil myristic acid C13H27, Cu-nutmeg, palm kernel, coconut oil palmitic acid C15H31, Cu-palm oil, animal and plant fats stearic acid C17H35, Cu-animal fat and plant oil arachidic acid C19H9, Cu-groundnut oil lignoceric acid C23H11, Co-groundnut oil unsaturated fatty acid unsaturated fatty acid are liquid at are character as Fatty acids having two or more double bonds are called polyunsaturated fatty acids, PUFA, while those with a single double bond are called as monounsaturated fatty acids, MUFA. Unsaturated fatty acids show different types of isomerisms. We have already learnt about the concept of isomerism in the last unit. You would realize that fatty acids with same molecular formula, as well as same number of double bonds, may differ in the location of double bonds and exhibit positional isomerism. Oleic acid has 15 different positional isomers. Again, the orientation of the two hydrogen atoms attached to the two carbon atoms joined by the double bonds may differ, geometric isomerism. If both hydrogen atoms are placed on the same side of the double bond, cis isomer results, but if these are placed on either side of the double bond, it is a trans isomer as shown in figure 2.4. Cis comes from Latin word meaning same or same side. Trans comes from Latin word meaning across or opposite sides. Examples of cis trans isomer is oleic acid and aladic acid. Nearly all naturally occurring unsaturated fatty acids are of cis configuration. Carbon atoms of fatty acids are numbered from carboxyl carbon. Various conventions are used to indicate the position of double bonds in the fatty acid molecules. Like, increment 9 indicates a double bond between carbon atoms 9 and 10 of the fatty acid. The Greek alphabets, alpha, beta, gamma, omega, are used to identify the location of the double bonds. The alpha carbon is the carbon next to the carboxyl group, and the omega is the last carbon of the chain because omega is the last letter of the Greek alphabet. Linoleic acid is an omega-6 fatty acid because it has a double bond 6 carbons away from the omega carbon. Similarly, alpha-linolenic acid is an omega-3 fatty acid because it has a double bond 3 carbons away from the omega carbon. In other words, fatty acids known as omega-3 or N3 fatty acids have one double bond between carbon atoms 3 and 4 from the omega carbon but no double bond between 6 and 7 carbon atoms. On the other hand, omega-6 or N6 fatty acids have double bonds between carbon atoms 6 and 7 from omega carbon. Table 2.2 presents the common unsaturated fatty acids found in nature. Table 2.2 Naturally occurring common unsaturated fatty acids. Look at the screen. While on the topic of fatty acids, we must also talk about essential fatty acids, EFA. Surely you must have heard about these important fatty acids. What are they? As the name suggests, EFA are essential to be provided in the diet. We must have them to live and to be healthy. Our bodies cannot make them from other substances. We must obtain an adequate supply from external sources, from food or from supplements. Two fatty acids are essential to human health. Can you name them? The first is the omega-6 EFA, which is called linoleic acid, LA. LA is abundant in polyunsaturated safflower, sunflower and corn oils. The second, known as the omega-3 EFA, is called alpha-linolenic acid, LNA, or ALNA. Sometimes referred to as super unsaturated, LNA is found abundantly in flax and hemp seeds. Look at Table 2.2 for the structure of these two fatty acids. LA and its derivatives belong to the omega-6 family of polyunsaturates. In addition to linoleic acid, La, this family includes gamma-linoleic acid, GLA, diamogamma-linolenic acid, DGLA, and arachidonic acid, R. LNA and its derivatives belong to an omega-3 family of supersterates. Besides alpha-linolenic acid, LNA, this family includes stearidonic acid, SDA, eicosapentaenoic acid, APA, and docosahexaenoic acid, DHA. If LNA is provided by foods, our cells make SDA, APA, and DHA. You will learn more about the EFA in the Advanced Nutrition course in Unit 5. Next, we shall focus on neutral fats in this unit, but before that, let us review what we have learned till now. 2.3.2 Neutral fats. Neutral fats are esters of fatty acids with glycerol and found abundantly in nature. These are insoluble in water but readily soluble in ether, chloroform, benzene and carbon tetrachloride. They are bland, odorless substances and neutral in reaction. Neutral fats are good solvents themselves for other fats, fatty acids etc. As glycerol has 3 hydroxyl groups, 3 molecules of fatty acids may get attached with it at the maximum. When one molecule of fatty acid binds with the glycerol, a monoacylglycerol, monoglyceride old name, forms. Diacylglycerol and triacylglycerol form when 2 or 3 molecules of fatty acids combines with one molecule of glycerol, as indicated in Figure 2.5. Fatty acids reacting with the glycerol may be same or different. Figure 2.5. Triacylglycerol. Look at the screen. From simple fats, we move on to compound lipids, wherein we shall focus on phospholipids. 2.3.3 Phospholipids. Phospholipids are a heterogeneous group of compounds widely distributed in animal tissues, particularly in the cell membranes. Phospholipids are classified on the basis of alcohol present in the molecule. These are classified as A. Glycerophosphatides, in which glycerol is the alcohol. B. Phosphoinositides, in which inositol is the alcohol. C. Phosphosphingosides, in which sphingosine is the alcohol. Glycerophosphatides are esters of fatty acid with glycerol containing an esterified phosphoric acid and a nitrogen base. Brain and nervous tissues, liver, kidney, pancreas and heart contain large quantity of phospholipids. In order to produce phospholipids, also called as phosphatides, first a phosphatidic acid is formed. In this compound, a phosphate group is present at the carbon atom 3, while 2 fatty acids are present in carbon atoms 1 and 2. This is, in fact, the simplest type of phosphoglyceride from which several other types are derived. The basic structure of phospholipids is very similar to that of the triacylglycerides, except that C3SN3 of the glycerol backbone is esterified to phosphoric acid as highlighted in the Figure 2.6. Figure 2.6. Structure of phospholipids. Look at the screen. Phospholipids exhibit important biological functions. They A. Increase the rate of fatty acid oxidation. B. Act as inorganic ion carrier across the membrane. C. Help in blood clotting. D. Act as prosthetic group for some enzymes. And A. Form the membrane structure. Some important phospholipids are phosphatidylcholine, lecithin, phosphatidylserine, phosphatidylethanolamine, cephalin, phosphatidylethanolamine, plasmalogen, diphosphatidylglycerol, cardiolipin, and phosphatidylinositol. The chemical structure of phosphatidylcholine, lecithin, illustrated here in Figure 2.7, is typical of the phosphatides found in the brain, lung and spleen. Look at the screen. Figure 2.7. Structure of lecithin. We learned earlier that fatty acids, steroids, hormones, vitamins A and D, etc. are categorized as derived lipids. You will find a detailed review of the chemistry of vitamins in Unit 3. Hence, we shall not dwell on them at this stage. Steroids are important compounds. We shall get to know the chemistry of these compounds next, followed by eicosanoids. 2.3.4 Steroids. Steroids form a group of compounds which are often found in association with fats, but structurally and functionally these are somewhat unrelated to most other lipids. Steroids are soluble in fat but resistant to sodium hydroxide, i.e. these are unsaponifiable. Basic structure of steroids consist of 3 cyclohexane rings, 1, 2, 3 rings, and 1 cyclopentane ring, ring 4, as in the Figure 2.8. The parent compound of steroids is called as cyclopentanoperhydrophenanthrene that has 2 methyl side chains typically at positions 10 and 13 constituting carbon atoms 18 and 19 in most steroids. Look at the screen figure 2.8, basic structure of steroids. Steroids are of many types. These are given below. A. Sterols, cholesterol, ergosterol. B. Bile acids, glycolic acid, taurocolic acid. C. Hormones, testosterone, estradiol, corticosterone. D. Vitamin D2 and D3A, cardiac glycosides, stropontin F, saponins, digitonin. Steroids containing an alcoholic group at C3 position and a side chain of 8 to 10 carbon atoms at C17 are called as sterols. The best known examples are cholesterol, yes, the much talked about compound because of its association with atherosclerosis, and ergosterol, which is a precursor of vitamin D, as illustrated in figure 2.9, A and B. Sterols, such as cholesterol, are alcohols with the cyclopentane ring system, atoms 1 through 17 in the figure 2.9a. This substructure is also found in steroid hormones such as testosterone, refer to figure 2.10, progesterone and cortisol. Cholesterol is considered an alcohol because it has a hydroxyl group, O, in position 3 of the ring system. While cholesterol is most abundant in the brain, nervous tissue, adrenals and skin, ergosterol is mainly found in yeasts, molds, etc. Sterols of vegetable origin are called phytosterols. They have the same basic structure as cholesterol, but differ in the side chains attached to carbon-17. Phytosterols, such as stigmasterol from soybean oil, are of current interest because they lower blood cholesterol levels. Look at the screen. A. Cholesterol, C27H45OH, B. Ergosterol figure 2.9, structure of sterols figure 2.10, structure of steroid hormone. After steroids, we shall focus on a family of compounds called eicosanoids derived from polyunsaturated fatty acids. What are eicosanoids? What is their role in the body? Let's find out. 2.3.5 Eicosanoids. Eicosanoids are the local hormones formed by body tissues during self-healing responses to stimuli. Eicosanoids are a family of compounds derived from polyunsaturated eicosanoic acids. They are produced from arachidonic acid, a 20-carbon polyunsaturated fatty acid. Eicosanoids comprise the prostanoids, leukotrienes, LTs, and lipoxins, LXs. Prostanoids include prostaglandins, PGs, prostacyclins and thromboxanes as shown in figure 2.11. You will realize that prostaglandins and related compounds are collectively known as eicosanoids. Let us learn about these different eicosanoids. Look at the screen figure 2.11. Classification of eicosanoids. Prostaglandins belong to a subclass of lipids known as the eicosanoids because of their structural similarities to the C20 polyunsaturated fatty acids, the eicosanoic acids. Prostaglandins are known to occur in almost all tissues in very small quantities and have important physiologic and pharmacologic activities. These are synthesized in vivo by cyclization of arachidonic acid, which is either derived from dietary linoleic acid or ingested as such, to form a cyclopentane ring. There are 3 fatty acids known as eicosanoic acids, characterized by the number of double bonds present in the structure, which by joining with the cyclopentane ring, gives rise to 3 groups of prostaglandins viz. PG1, PG2, PG3. These are listed in table 2.3. Name molecular formula. Position of unsaturation food source timnoionic acid C19H33, COOH5, 8, 11, 14, 17 fish oil glupanodonic acid C12H32, COOH7, 10, 13, 16, 19 fish oil cervonic acid C21H31, COOH4, 7, 10, 13, 16, 19 fish oil. Variations in the substituent groups attached to the rings give rise to different types in each series of prostaglandins. Of the 6 types of prostaglandins known, the primary types are prostaglandins A and F. All prostaglandin A types have a keto group in position 9 of the structure, but all prostaglandin F types have a hydroxyl group in that position. Thromboxanes are the second group of eicosanoids, first discovered in platelets and found to have a cyclopentane ring in the structure that is interrupted with an oxygen atom. Different types of thromboxanes, A, B, etc., in each series, 1, 2, 3, occur due to the variation of the substituent groups attached to the ring. Leukotrienes are the third group of eicosanoids, first discovered in leukocytes. These are characterized by the presence of 3 conjugated double bonds. Figure 2.14 presents the structure of leukotriene. In the section above, we learnt about the lipids, their classification and structure. This basic knowledge, you would realize, will be useful to us in understanding the chemical properties of lipids, particularly the fatty acids and neutral fats, which is the focus of the next section. 2.4 Chemical properties of fatty acids and neutral fats. Fatty acids and neutral fats respond to different chemical reagents in different ways depending on the nature of these substances. Some of the important reactions of both of these groups of compounds are discussed briefly here. We start with the chemical properties of fatty acids. 2.4.1 Chemical properties of fatty acids. This section is a review of the chemical properties of fatty acids, which include esterification, hydrogenation, halogenation, etc. Recognition of these properties is useful in understanding fatty acids. Let us learn about these properties then. Esterification. Like any other organic acid, fatty acids also form esters with various alcohols. An alcohol such as glycerol is reacted with fat or oil to produce esters such as mono and diacyl glycerols. Using the esterification process, edible acids, fats and oils can be reacted with edible alcohols to produce useful food ingredients that include many of the emulsifiers such as mono and diglycerides, lecithin etc. Soap formation. When fatty acids react with alkalis, metallic salts of fatty acids commonly called as soaps are formed. Potassium soap of fatty acids is more water-soluble than sodium soap. Hydrogenation. When exposed to hydrogen at high pressure and temperature in presence of Ni or Pt catalyst, an unsaturated fatty acid, containing a double bond, accepts the hydrogen at the double bonds and is converted to a saturated fatty acid as shown herewith. Generally speaking, hydrogenation is used to change liquid oil into a semi-solid or solid fat at ambient temperatures to enhance oxidative stability. Halogenation. Fatty acids accept chlorine and iodine at the double bonds when treated with reagents such as iodine monochloride and a fatty halide results. 2.4.2 Chemical properties of neutral fats. The chemical properties of neutral fats are highlighted in this section. These include saponification. Fats when boiled with alcoholic solution of NaOH or Co undergo hydrolysis into glycerol and fatty acids and the latter form soaps with Na or K. The reaction is known as saponification. Hydrolysis. Fats when boiled with water at 220 oC under pressure in an autoclave, undergo hydrolysis to first form a diglyceride and then ultimately glycerol and fatty acids are formed as illustrated in the figure 2.16. Look at the screen. Figure 2.16. Hydrolysis of a fat molecule to yield glycerol and fatty acids. Hydrogenation. We have already talked about the hydrogenation property of fats in subsection 2.4.1. Hydrogenation, you learnt, is the process of turning liquid oil into solid fat. In presence of finely ground Ni or point catalyst and at 150 to 220 oC, glycerides of unsaturated fatty acids can be saturated by the action of hydrogen. Partial hydrogenation produces margarines, shortenings, shortening oils, and partially hydrogenated vegetable oils. These products contain large quantities of trans fatty acids and other altered fat substances, some of which are known to be detrimental to health because they interfere with the normal biochemical processes. Trans fatty acids are considered even more harmful than saturated fatty acids. Rancidity. This results from the formation of aldehyde due to the oxidation of unsaturated glycerides or by the liberation of fatty acids due to hydrolysis. In autoxidation, oxygen reacts with unsaturated fatty acids. Initially, peroxides are formed, which in turn break down to hydrocarbons, ketones, aldehydes and smaller amounts of epoxides and alcohols. The result of the autoxidation of fats and oils is the development of objectionable flavors and odors characteristic of the condition known as oxidative rancidity. 2.5 Chemistry of proteins and nucleic acids. Proteins are the third group of macronutrients and much attention should be given to these extremely important compounds. Nucleic acids are the main constituents of gene and are responsible for protein synthesis. You being a student of dietetics should have some idea of such an important group of compounds that controls our activities. This subsection will focus on the following aspects of protein and nucleic acid chemistry: definition of amino acids, peptides, proteins and nucleic acids, classification of amino acids, peptides and proteins, structure of proteins, physico-chemical properties of amino acids and proteins, biologically important peptides, structure and classification of nucleic acids. We begin our study on this topic by first getting introduced to proteins and nucleic acids. Introduction to proteins and nucleic acids. Protein is derived from a Greek word, protos, which means preeminent, first or foremost, or primary matter. This name was suggested by Berzelius to Dutch scientist Gerard Johannes Mulder, who coined the term protein in 1838. Earlier, protein was described as albuminous material, in that it resembled albumin or the white of egg. Proteins are compounds of carbon, hydrogen, oxygen and nitrogen. On an average, proteins contain 16% nitrogen. Most proteins also contain sulfur and some proteins contain iron, copper, phosphorus and zinc. Proteins are, in fact, polymers consisting of chains of monomeric units. The chains are essentially linear and contain no branches. The monomeric units of proteins are amino acids, which you may already know are the building blocks of the body, and the linkage between two amino acids is called a peptide bond. We will learn about the amino acids and the peptide bond in a little while from now. A typical protein contains about 300 amino acids and proteins have a molecular weight ranging from 5,000 to several million daltons. Proteins are essential constituents of living organisms and found in all plant and animal foods. Next, let us get to know what are nucleic acids. Nucleic acids, like proteins, are high molecular weight polymers present in all living cells. The two major classes of nucleic acids are DNA and RNA. Surely, you are aware of these nucleic acids. The monomeric units of nucleic acids are called as nucleotides. Complete hydrolysis of nucleic acids yields heterocyclic nitrogenous bases, 5-carbon sugars and phosphoric acid molecules. Their main function is storage and transmission of genetic information. With a basic introduction to proteins and nucleic acids, let us get to know more about amino acids, proteins and nucleic acid with respect to their structure, classification and properties. We shall begin with amino acids. 2.6 Amino acids, structure, classification and properties. Amino acids are the building blocks of proteins. All proteins consist of a sequence of amino acids, which are compounds containing an amino group, NH2, and a carboxyl group, COO, as indicated in the general structural formula in the figure 2.17. Normally 20 different 2-amino acids or alpha amino acids having NH2 group at carbon 2, are found in proteins, though very recently 2 more amino acids are also found to be present in some proteins. All the amino acids can be represented by a general structural formula, in which R, the side chain, is a variable factor that changes from amino acid to amino acid, giving a character specific to a particular amino acid. 2.6.1 Classification of amino acids. You would realize that amino acids are classified into 4 broad groups according to the nature of the side chain, R group. These are A. Amino acids with non-polar side chain. B. Amino acids with polar but uncharged side chain. C. Amino acids with positively charged side chain, and D. Amino acids with negatively charged side chain. Let us get to know about them. 1. Amino acids with nonpolar or hydrophobic side chain. These are 8 in number, as indicated in figure 2.18, alanine, leucine, isoleucine, methionine, phenylalanine, proline, tryptophan and valine, ala, al, lu, met, phe, pro, trp and val. These are sparingly or less soluble in water than polar amino acids. Hydrophobicity increases with an increasing side chain length. Look at the screen figure 2.18, structures of amino acids with hydrophobic side chain 2. Amino acids with uncharged polar, hydrophilic, side chain. These have neutral polar functional groups and are able to establish hydrogen bonding with appropriate molecules, such as water. Polarity of serine and threonine is due to O group and of asparagine, ASN, and glutamine, GLN, is due to CONH2, amide, groups. Sometimes glycine is also included in this group. Cysteine and tyrosine possess most polar functional groups of this category, since both thiol and phenol groups may undergo partial ionization at pH values close to neutrality. In proteins, cysteine is often present in oxidized forms, i.e. cysteine. Asparagine, ASN, and glutamine, GLN, also readily hydrolyze to form aspartic acid, ASP, and glutamic acid, GLU, in presence of an acid or alkali. Figure 2.19, structures of amino acids with hydrophilic side chains 3. Amino acids with positively charged side chains, at pH close to neutral, lysine, histidine and arginine are the examples of this group. Amino group of lysine and guanidino group of arginine are responsible for plus-way charges. Imidazole group of histidine is 10% protonated at pH 7.0 and 50% at pH 6.0. Figure 2.20, structures of amino acids with positively charged side chains 4. Amino acids with negatively charged side chain aspartic acid, ASP, and glutamic acid, GLU, are the examples of this class. Figure 2.21, Structures of amino acids with negatively charged side chains. For your convenience, a complete list of these 20 amino acids is presented in Table 2.4. Amino acids with non-polar side chain: alanine, valine, leucine, isoleucine, proline, methionine, phenylalanine. Tryptophan. Amino acids with polar but uncharged side chain: glycine, serine, threonine, cysteine, tyrosine, asparagine, glutamine. Amino acids both positively charged side chain: lysine, arginine, histidine. Amino acids with negatively charged side chain: aspartic acid, glutamic acid. With this, we come to an end of our discussion on classification of amino acids. Next, let us learn about the chemical reactions of amino acids. But first, let us recapitulate what we have learned so far about amino acids. 2.6.2 Properties and chemical reactions of amino acids. In this section, we shall look at the physical and chemical properties of amino acids. We shall first study about the physical properties of amino acids. A. Physical properties. Solubility. Amino acids are readily soluble in water, slightly soluble to insoluble in ethanol, and insoluble in ether. Tyrosine is soluble in hot water but only sparingly soluble in cold water. Cysteine with difficulty is soluble in only hot water. Proline and hydroxyproline are soluble in alcohol and ether. Amino acids are generally soluble in acids and bases and form salts. Tyrosine is moderately soluble in acids and bases. Cysteine is soluble in strong mineral acids, HCl, but slightly soluble in acetic acid and dilute ammonia. Melting points. Amino acids have a high melting points in the range of 200 to 300 degrees Celsius. Taste. Amino acids are usually sweet, tasteless or bitter. E.g. Geely, Ala, Val, Pro, Hydroxy-Pro, Sir, Tri and His are sweet. Lu is tasteless, whereas Il and R are bitter in taste. Sodium salt of glutamic acid, monosodium glutamate, MSG, also known as Ajinomoto, is valuable as a flavoring agent for certain foods and sauces since it imparts and enhances the flavor, also for meat and meat products. MSG is being used since 1908 when a Japanese chemist Kikuni Akeda discovered that MSG could improve the flavor of the food products. However, tests on animals have shown that it can cause brain damage and cancer. It has not been proved to be dangerous to humans. However, food experts do not approve its use for infants and pregnant women. The acceptable daily intake of MSG for a person weighing about 60 kilograms is approximately 7.2 grams. Acid-or-basic properties. Depending upon the pH of the solution, amino acids ionize and act as weak Bronsted acid or base, i.e. proton donor or acceptor. This ionization facilitates their quantitative analysis. Thus, all the amino acids may be an acid or a base. Let us understand this concept better. Look at the structure of amino acid given in Figure 2.22. It is evident that amino acids contain both acidic Cu and basic NH2 groups. Because of this unique structure, amino acids have the characteristics of an acid and a base and are capable of reacting chemically either as an acid or a base. Hence, they are said to be amphoteric. They can donate or accept protons and thus can serve as an acid or a base. However, when both the groups are ionized, the amino acid is said to be a Switterian and behaves as a neutral compound. Among amino acids, neutral amino acids have a net zero charge because their structure is as illustrated in Figure 2.23. The positive and negative charge neutralizes each other. Any compound which has a net zero charge is called a Switterian. Switterons are amphoteric molecules, no net charge on the molecule, which behave both as acid and base, and the pH at which this form exists is known as isoelectric point, pi. Stereochemistry of amino acids. Optical properties. All amino acids, except glycine, rotate the plane of polarized light because of the presence of an asymmetric center at C2. Both L and D enantiomers are possible for the amino acids. What do we mean by enantiomers? Do you recall reading about them in the last unit under the topic of isomerism? Like carbohydrates, amino acids too exist as L and D isomers. The definition of D and L depends on the position of NH2 group on C2. When the amino group is on the left of C2, it is called L form. Essentially, all the amino acids in the diet and in the body occur as the L isomer EL, THR, hydroxyproline and hydroxylysine have second asymmetric carbon also in their structure. Thus 4 stereoisomers are possible for these amino acids. Optically active amino acids can be converted into racemic mixture by heating them with alkali. Hydrophobicity of amino acids. Hydrophobicity of amino acids and also of peptides and proteins may be determined by relative solubility of amino acid in water and in a less polar solvent, e.g., ethanol, respectively. Amino acids are grouped as hydrophilic and hydrophobic depending on whether the side chains are, like water, hydrophilic, or hate water, hydrophobic. ALA, R, ASN, asparagine, ASP, CYs, GLU, GLN, glutamine, GLI, HIS, LYS, PRO, SIR, THR are classified as hydrophilic. Sometimes, TYR is included in this group, though TYR is more hydrophobic than hydrophilic. Amino acid side chains other than above are hydrophobic. Absorption spectra and fluorescence properties of all the amino acids, TYR and Phe, absorb UV light and have a maximum absorption at 280, 274 and 260 nanometers, respectively. Cysteine shows a slight absorption at 238 nanometers. All the amino acids absorb at wavelength near 210 nanometers. TYR and Phe are the only amino acids which show natural fluorescence. Trifluorescence remains even when it is bound in protein. Next, let us get to know about the chemical properties of amino acids. B. Chemical reactions of amino acids. The chemical reactions of amino acids can be divided into three classes. I. Reactions of carboxylic group. E. Reactions of amino group. E. Reactions of the side chain. Here in this unit, we shall not go into each and every reaction specific to the three classes mentioned above, but few important reactions are described herewith. I. Reactions of carboxylic group. A. Salt formation. In alkaline medium, COOH group reacts with metal hydroxide to form amino acid salts as shown in figure 2.25. Figure 2.25, salt formation. B. Ester formation. In presence of dry HCl, amino acids react with alcohol to form esters. This is one of the ways of blocking COOH group in the chemical synthesis of proteins. Figure 2.26 illustrates the ester formation. Figure 2.26, two-step reaction showing ester formation with alcohol. This ester as well as free amino acid can be reduced to corresponding alcohol with lithium aluminium hydride, Li-Al4. This is one of the ways of identifying C-terminal amino acid in the form of amino alcohol. C. Decarboxylation. Amino acids undergo decarboxylation reaction, enzymatically or by treatment with heat, acid or alkali, barium hydroxide, to form the corresponding amines as shown in figure 2.27. Figure 2.27, decarboxylation reaction in biological systems. A number of amines generated by this way have biological activity. For e.g., histamine, decarboxylation product of histidine, mediates allergic reactions, shock and stress. Similarly, histamine and tyramine, decarboxylation product of tyrosine, both possess pharmacological properties. E. Reactions of amino group. There are many reactions of amino group, namely methylation, reaction with nitrous acid, oxidative deamination of amino acids, reaction with aldehydes, etc. We shall not discuss each one of them. Some important reactions are described here. Ninhydrin reaction. Ninhydrin is a powerful oxidizing agent. When it reacts with amino acids, oxidative decarboxylation results in the formation of CO2, NH3 and an aldehyde. The reduced ninhydrin subsequently reacts with liberated NH3 forming a blue-purple complex which has maximum absorption at 570 nanometers. Proline forms a yellow color in this reaction. This reaction is very useful for quantitative estimation of amino acids. Figure 2.28, ninhydrin reaction. Sanger's reaction. This reaction is specific for the amino group of the amino acids or peptides. 1-FLURO2, 4-dinitrobenzene, FDNB, reacts with the sample resulting in the formation of DNP derivative, which is a yellow crystalline compound. Figure 2.29, Sanger's reaction. Edman's reaction. This is an important reaction for the identification of the amino acid at the N-terminal of the peptide. Phenylisothiocyanate reacts with the amino acid to yield a cyclic product, phenylthiahydont 1.